Hello everyone, welcome to my channel Guidental. This is Dr. Saib Singh. So the subject for today's video is going to be biochemistry. Whatever topics I'll be mentioning in the video are also mentioned in my blog by the name of Guidental, the link for which is given in the description of the video. So starting with the first chapter, that is carbohydrates. In carbohydrates, you should know the classification of carbohydrates, that is into simple and complex carbohydrates in detail with examples. Isomerism, you should know the definition. Types, that is optical, functional and stereoisomerism with examples. Reactions of monosaccharides, you should know the definition and examples. Tautomerization, oxidation, reduction and oxazone formation. In reduction, a special short note could be asked to you about the difference between reducing and non-reducing sugars. Some important sugars, you need to know the structure, that is the component sugars. Isomerization if applicable, bonds or linkages if applicable, and any one used for glucose, fructose, galactose, sucrose, maltose, lactose, cellulose, inulin, glycogen, starch and hyaluronic acid. Coming to carbohydrate metabolism, you should know the difference between oxidative phosphorylation and substrate level phosphorylation. In pathways, study under definition, site of occurrence, cycle or steps, ATP yield and clinical significance for glycolysis, HMP shunt, uronic acid pathway, Cori cycle, citric acid cycle, gluconeogenesis, glycogenesis and glycogenolysis. In glycolysis, you should know the difference between glucokinase and hexokinase. Coming to glycogen storage diseases, the types, that is 1 to 8 types, and the name of defective enzyme in each. Only this, that is which type and which enzyme is defective in each of the types. Only that you need to know. Coming to galactose metabolism, you should just know the difference between non-classical and classical galactosemia. Next chapter is proteins and amino acids. You should know that protein is the only source of nitrogen to the body. That is, protein contains about 16% nitrogen by weight. In amino acids, you should know the basic structural formula. Classification you should know according to metabolism, that is purely ketogenic, purely glucogenic and ketogenic and glucogenic, and according to nutritional requirements, that is essential, non-essential and semi-essential with examples. Coming to properties of amino acids, you should know about the isoelectric point and peptide bond. In proteins, you should know the structure of proteins, primary, secondary, tertiary and quaternary structure of proteins with examples as well as diagrams. And the denaturation of proteins, you should know the definition and the clinical significance. In protein metabolism, you should know about transamination, deamination and transdeamination, that is the definition, types if applicable, enzymes involved, any one clinical significance and any one example in each. Coming to urea cycle or Krebs-Henseleit cycle, you should know the definition, site of occurrence, cycle, ATP use, yield and the clinical significance of in the urea cycle. An important MCQ that could be asked: creatine phosphate is the storage form of energy in muscle which is broken down to phosphate to release energy in the form of ATP. This is known as Lohmann's reaction. Next topic is phenylalanine and tyrosine metabolism. You should know the product synthesized, that is melanin, catecholamines or epinephrine and thyroxine. Disorders of tyrosine metabolism or inborn errors of metabolism. You should know about the enzyme deficient and the clinical features regarding phenylketonuria, alkaptonuria, albinism, maple syrup urine disease and Hartnup disease. Next chapter is lipids. You should know the classification of lipids, that is simple, complex and derived with any one example in each. Classification of fatty acids according to saturation, that is saturated and unsaturated in detail with subtypes and examples, and the concept of saponification and saponification number. Now, coming to classification of lipoproteins, you should know about the clinical significances of chylomicrons, VLDL, LDL and HDL, that is very low density lipids and high density lipids. Coming to lipid storage disorders, you should know the enzyme defect and the lipid type accumulated regarding Gaucher's disease, Niemann-Pick disease, Fabry's disease, Tay-Sachs disease and Farber's disease. Coming to metabolism of lipids, you should know about the beta oxidation of palmitic acid in detail. Alpha oxidation of fatty acids, just know the difference between the beta oxidation and the clinical significance. In ketogenesis, you should know the site of occurrence, cycle, primary ketone bodies, that is acetoacetate, secondary ketone bodies, that is beta hydroxybutyrate and acetone, and the names of major enzymes involved, that is HMG, coenzyme A lyase, and the clinical significance. Coming to ketolysis, you should know the site of occurrence and the clinical significance. In ketosis, you should know the normal level of ketone bodies in blood, that is less than 1 milligram per deciliter, and the clinical features. Cholesterol, you should know the clinical significance, site of synthesis. Acetyl coenzyme A is the building block of cholesterol. This is an MCQ that could be asked in biochemistry. Coming to bile acids, you should know the clinical significance, site of synthesis, types, that is primary and secondary bile acids, with examples. Next chapter is or topic is biological oxidation or electron transport chain, that is ETC. You should know the definition and the site of occurrence, names and functions of the complexes involved, concept of respiratory chain, concept of ADP O or PO ratio, and the ratio for NADH and FADH2, ATP synthase or complex 5 of ETC, that is the functions of FO and F1 subunits, and the clinical significance of ETC. Next topic is liver function test or LFT. You should know the indications of LFT, any 4 to 5, classification, that is liver's capacity to metabolize drugs, serum and urine bilirubin and urobilinogen, hepatocyte injury tests, tests of liver's biosynthetic capacity, serum bilirubin, urine bilirubin, urobilinogen, and amino transferases, alkaline phosphatase, serum albumin and ceruloplasmin. The subtopics that are to be discussed in all of these enzymes or the parameters of liver function test, these are mentioned in my blog in detail, so you can refer the blog for further details, because it is the same repetition for each point again and again, so it is better if you refer the blog. There I have mentioned exactly what points and what parameters have to be studied in detail. Now, an important MCQ in liver function test, which is very important, that is ALT, that is alanine transferase, is more specific than AST, that is aspartate transferase. Next topic is kidney function test. You should know the indications of kidney function test, any 4 to 5, and the classification. Classification, you should know there are tests to evaluate the glomerular filtration rate and the tests to evaluate tubular function. The normal values of serum and clearance values of creatinine, urea, and inulin with names of any 2 to 3 conditions associated with abnormal clearance, and increased or decreased values in case of serum creatinine, inulin, and urea. In urine analysis, you should know the color of urine, that is name of any one abnormal condition associated with red, yellow, black, and brown color of urine. Volume of urine, that is normal value of urine output. Names of conditions associated with anuria, oliguria, and polyuria. Glucose in urine is seen in diabetes, and ketone bodies in urine is seen as diabetic ketoacidosis and starvation. And lastly, you should know the causes of increased and decreased BUN or serum creatinine ratio. The next topic is vitamins and minerals. In vitamins and minerals, you study the following vitamins under chemical name, recommended daily allowance, sources, functions, and deficiency diseases of vitamin A, B, C, D, E, and K. In synthesis of vitamin A and D is important, that is the synthesis of vitamin D, and the A is also known as Waltz or rhodopsin synthesis. Study under RDA, sources, functions, deficiency, and excess disorders of sodium, potassium, calcium, fluorine, and iodine. The next chapter is enzymes. You should know that all enzymes are proteins except the ribozymes. Classification of enzymes, study under function of the class of enzyme and any 2 examples each. A mnemonic to remember the classification of enzymes is OT-HIL. O is oxidoreductases, T is transferases, H is hydrolases, I is isomerases, L is lyases, and again L is ligases. You should know the definition and examples of holoenzyme, apoenzyme, coenzyme, active site, and metalloenzyme. The factors affecting enzyme activity, you should know the concentration of enzyme, Michaelis-Menten constant, that is the concentration of substrate, temperature, pH, and time. Coming to mechanism of enzyme action, you should know about the enzyme substrate complex theory, lock and key model, and induced fit model. In enzyme inhibition, you should know the differences between competitive and non-competitive inhibition, suicide inhibition, and allosteric inhibition with any 2 examples each. Isoenzymes, you should know the definition and examples, that is glucokinase and hexokinase. So last topic for biochemistry is nucleic acids. You should know the concept of nucleosomes, linker DNA, nucleotides, nucleosides, purines, pyrimidines. These all topics, the nucleic acids and the enzymes, they have already been taught to you in 11 12. It is the same repetition of your 11 12 knowledge in biochemistry. So coming to nucleic acids, you should know the concept of nucleosomes, linker DNA, nucleotides, nucleosides, purines and pyrimidines, the central dogma of life, Watson and Crick model of DNA in detail with diagram, along with Chargaff rule and hydrogen bonding, definitions of bDNA, zDNA and aDNA and where they are seen, DNA replication in detail, RNA you should know the types, that is percentage, site of presence and function. Study the clover leaf or hairpin loop model for tRNA, that is transfer RNA. Transcription, post-transcription modification and translation are to be studied in detail. Operon and lag operon model can be asked in your short notes. So thank you so much for watching the video. This is all that has to be studied, the important topics that need to be studied for the subject of biochemistry. If you found this video to be useful, please like, share and subscribe to my channel Guide Dental for further updates. Thank you so much.